Hello everyone, my name is John Francis, and I'll be talking about the relationship between cholinergic and noradrenergic neuromodulatory activity in behavioral state in mice. This poster demonstrates my honors thesis satisfying requirements for departmental honors from the biology department and requirements from the Robert D. Clark Honors College. An animal's ability to engage with a particular task is highly variable. However, there exists an optimal arousal state under which an animal can perform optimally. Arousal encompasses both brain state, which describes the physiological activity of the brain, and behavioral state, which describes observable behavioral patterns and movements of the animal. Moment-to-moment fluctuations in brain state and arousal ultimately influence an animal's ability to perform and engage in different perceptual tasks. These moment-to-moment fluctuations in behavioral state and arousal are influenced by various neuromodulatory systems within the brain. Neuromodulators are neurons that modulate the activity of other neurons. Varying activity within these neuromodulatory systems produce changes in an animal's cognition and sensory processing. Two neuromodulatory cell types studied in my project are cholinergic and noradrenergic neurons, which produce and release the neurotransmitters acetylcholine and noradrenaline, respectively. Cholinergic projections from nuclei in the basal forebrain and noradrenergic projections from nuclei in the locus coeruleus innervate to various target regions of the cortex. Previous literature also shows that changes in neuromodulatory activity are linked to changes in arousal state and behavior. Given this background, I address the following question. What is the relationship between cholinergic and noradrenergic neuromodulatory activity and behavioral state with respect to diverse arousal and behavior-dependent modes of neuromodulation? To answer this question, we first had to visualize cholinergic and noradrenergic axon activity in awake mice. GCAMP6S, a fluorescent calcium indicator, was injected into the basal forebrain of chat mice to allow for cholinergic axon visualization. Because of the transgenic gene expression of DBH mice, viral injection of GCAMP6S into the locus coeruleus of DBH mice was not required for noradrenergic cortical activity visualization. Following viral injections and cranial window implantation, simultaneous 2-photon calcium imaging and behavioral data acquisition was performed to directly compare axonal activity to behavioral state. After performing simultaneous 2-photon microscopy and behavioral recording, we show that ACh, shown on the left, and NA, shown on the right, neuromodulatory related axonal fluorescence, is closely related to moment-to-moment changes in behavioral state. As indicated by the red dashed boxes, increases in whisker motion energy, or walk velocity, were both accompanied by increases in pupil diameter and by synchronous increases in both ACh and NA axonal DF over F, a measure of changing fluorescence, across all regions of interest, with each region of interest being represented by a different color. To quantitatively determine the temporal differences in cross-correlation between neuromodulatory activity and behavior, the peak cross-correlation across all chat and DBH mice were averaged, with the ACh max cross-correlation with behavior being depicted on top and DBH on the bottom. We see that the averaged ACh and NA max cross-correlation with behavior preceded the onset of whisking and followed the onset of walking. Additionally, we see a statistically significant difference between these two values, indicating varying neuromodulatory timescales across behavioral states. Last, to determine the relationship between axon-to-axon cross-correlation and behavioral state, the max cross-correlation between axons for all chat and DBH mice recordings were averaged for all periods of stillness, whisking, and walking. And for the cholinergic data on top, we see that the max cross-correlation between cholinergic axons was highest during stillness, intermediate during whisking, and lowest during walking. Additionally, there's a statistically significant difference between these three groups, providing evidence of local specificity across behavioral states. However, for noradrenergic data on the bottom, we see that the max cross-correlation between noradrenergic axons did not differ by behavioral state. And so in the noradrenergic neuromodulatory system, we do not see this local specificity across behavioral states. Future work on my project includes identifying the specific regions of the cortex from which these regions of interest were recorded. This would potentially provide us with a better understanding of the relationship between neuromodulation and multimodal cortical interactions. Additionally, future work also includes implementing other behavioral motifs into our behavioral analyses, such as snout movement, facial twitching, grooming, and tail motion. This would provide us with a more comprehensive understanding of neuromodulatory signaling with respect to more varied behavioral and arousal states, and, in turn, be more reflective of the mammalian brain. Thank you to my mentors Dr. David McCormick and Dr. Lindsay Collins for their guidance on this project, and thank you all for listening.